Welcome to Chapter 7, Part 2, where we are continuing to talk about membrane proteins and the function of transporters and channels and a couple other examples. As we mentioned, there are no fewer than 6 different major functions, and many proteins have more than one. In this chapter, we're really going to focus on these two right here. Cell-cell recognition and transport. Signal transduction we'll talk about later on. Anchorage and some of these other ones you might see in other classes or you might see in physiology down the line. But for now, we're going to focus on cell-cell recognition and transport. And don't forget that when we talk about transport, we can have unregulated diffusional transport. We can have facilitated diffusion in the form of channels or facilitated diffusion by carriers. Certain carriers can also hydrolyze energy molecules like ATP in order to do active transport to move things against their concentration gradient. Over here we have just basically a very specialized channel known as aquaporin. These are the ones that allow rapid equilibration of water across the plasma membrane, more so than letting other things through. We let through water quite rapidly. To show you how important this can be, there is a type of chloride channel. This is a channel that allows chloride ions to move across the plasma membrane. They have a negative charge, they're an anion. And under normal situations in your airway, chloride can move to the outside surface of the airway, and that helps bring water. And that water will then dilute the proteins and other things in mucus and other material and create a nice smooth lining in the airway. In cystic fibrosis, what happens is the person has a mutant CFTR that cannot move chloride. When you can't move chloride, the water can't get to the surface and the mucus still is secreted, but it becomes very, very viscous. Your airways get very, very small. If you've ever had a stuffy nose, you know what it's like to have viscous mucus in an airway, but imagine an airway 1/100th that size with that. And that's what's happening almost all the time with folks who have cystic fibrosis. It was not too long ago that the average age of a person before they passed away from cystic fibrosis was in their early to mid-20s. It's gotten better. We've gotten better therapies, but it's still a very, very significant thing just due to one mutation in this one channel. A major aspect of cell biology is regulating the concentration of molecules on one side or the other of a membrane of some kind, or regulating them in 3 dimensions. One of the reasons why is that when you have a concentration, those molecules will want to move to an area of lower concentration, and we can actually harvest energy from that. It is possible to harvest energy from molecules that are diffusing down their concentration gradient. When we get to ATP synthesis, you'll see that in spades. In any case, molecules want to move and spread out. And so we call that movement to spread out into areas of lesser concentration diffusion. And this will happen on its own, like for instance in this artificial situation where we have a membrane with pores in it and we have some food coloring on one side, and what will happen is some will get through and some won't, but eventually what will happen is we will see the molecules reach what we call equilibrium, where they're moving across the membrane as fast as they're moving back across the membrane. So when we have net diffusion, that means we have to have a net gradient. So diffusion is moving this way as molecules move through. They're still moving this way, but by the time the concentrations are equal, then they have achieved equilibrium. Now, a cell will usually not let this happen. A cell will usually do something to keep diffusion moving in a certain direction. But that's for another time. I want you to understand why we spend so much time on diffusion. Let's add a second solute this time. So now we've got the dye on one side and we've got another dye on the other side. And what we would see is a net diffusion of the orange and a net diffusion of the purple in the opposite direction until such time that they reach equal concentrations on both sides. And now we have equilibrium. And again, a cell will set this up. It will create this, but will never or hardly ever allow this. When we're talking about diffusion, whether it's pure diffusion or facilitated diffusion, what we have going on is the substances are moving from a higher concentration to a lower one. If water is doing it, we call that osmosis. But water is special, and I'll explain why later. We're not investing energy at this point. In fact, if anything, cells will take advantage of the energy inherent in that gradient in order to harvest and couple it to energy requiring reactions. But it's passive because no energy is expended during that time to make it happen. And we have simple diffusion. That's just where things can move through the plasma membrane. But generally what we're talking about there are very lipophilic, very small molecules. They're not many. Gases, occasionally gases can do this. But we do have channels that allow this, and we do have carriers. Channels are like protein tubes that are restricted by size and charge. Carriers can be much more diverse, but they're much more like enzymes because you have a binding, change in conformation and release. But because we're not investing energy in the process of these molecules moving, it's considered passive transport. Facilitated, but passive. Now, I promised you a few slides back that I'd talk about osmosis, and the lovely definition here. Osmosis is the passive transport of water across a selectively permeable membrane. That's great. But what does it mean? What does it mean? It means water is diffusing down its concentration gradient. Okay. One of the things I wish that we could do more of in these types of classes is to have you focus on the meaning rather than the simple repetition of the words. Sometimes repetition is good. There's nothing wrong with repetition in and of itself, but it doesn't convey meaning sometimes. And that's where the real action is in the meaning. The water is moving down its concentration gradient. So over here on the right, we have a higher concentration of water than we have on the left. And how do we know that? Because there's more solute on the left. There's more solute. And so water wants to move down its concentration gradient until we have roughly the same concentration of solute on both sides. But here's a really, really cool thing. It will diffuse across until the force of gravity or some other oppositional force prevents it. This is literally the interface of chemistry and physics. This is so cool because you can measure this as a physical force. You can put a little transducer here that can measure pressure, and as water is coming across here, it'll push across it, and you can measure it just like you were weighing a watermelon. Okay? That's the beauty of osmosis. It's this interface between physical forces and chemical forces. But it's diffusing across this membrane until either the concentration is equal on both sides or the force against the diffusion overcomes the diffusion itself. Really, really cool, and it's missed by a lot of people. And so here's that experiment where you can see water diffusing this way, and after a while you'll see a higher water level, and it will rise until the force of gravity prevents that diffusion, or if it hits the edge of the container. If it hits the edge of the container, though, the wall of the container is pushing against that solution too. It's just another kind of force. The water is moving into an area of lower concentration, and that's usually the opposite of solute. Solute's usually moving down its concentration gradient. If it's allowed to move, it may not be allowed to move. Water, if it's allowed to move, will move down its concentration gradient. Now you remember I said that physical force that osmosis will impart on the container or on the walls around it, that physical force can cause a cell to get bigger or to shrink. And that phenomenon we call tonicity. Now you may learn about tonicity in chemistry, and it's similar, and it is the same thing, but there's a different emphasis here. In biology, in cell biology in particular, tonicity is talking about the impact of that solution on a cell. So if you have a cell and you bathe it in an isotonic solution, what will happen is there's no net movement of water because the osmolarity on both sides is identical. The other possibility is that water can't move and the solutes can't move. There are water impermeant membranes out there in the world. A classic example of a water impermeant membrane is your skin, okay? or your large intestine is a relatively water impermeant membrane. Isotonic means that there is no net exchange of water across a given plasma membrane. Okay, it's the impact on the cells. A hypertonic solution means that there is a net loss of water out of the cell, usually because the solute concentration is greater than inside the cell. Okay, that's because there's more solute, less water means the water is going to leave the cell and go out to where it's diffusing out into its lower concentration. And then the opposite, a hypotonic solution. I like to think hypo, it's swelling, okay? Hypotonic means that the solute concentration inside the cell is less and the cell is gaining water. There are some complications here, and when we get into physiology in 330, we're going to show you osmolarity and tonicity and how they're not always exactly the same. And animal cells, because they have aquaporins, if they're put into a hypotonic situation, they will burst after a few seconds or minutes. That's because they just don't have any way to counteract that osmotic force. Plants, on the other hand, can do quite well in hypotonic situations because they have a cell wall. And so what happens is the water moves in until the water is pushing against the cell wall and the wall is pushing back, and so you can't get any bigger. That's why I made a point in that earlier slide about the idea of the walls pushing back against the container and preventing that osmotic force from taking place. Isotonic, red blood cells are perfectly happy there. They're exchanging water at an equal rate, so they will retain their original shape. Plants can do okay in isotonic situations, but they oftentimes will begin to lose that pressure that they need to stay upright. So like non-woody plants oftentimes will become a little wilted, and that's what we mean by flaccid. And then if we have a hypertonic solution, the water will move out of the red blood cell and it will crenate. That's another word for shrivel. A crenated cell is a shriveled cell. And what happens in a plant cell is that the water moves out of the cell. The cell wall stays the same, but the cell itself begins pulling away from that cell wall. This is a dangerous time for a plant. If it doesn't have compensatory mechanisms, it could die because it really does depend on being sort of adjacent to that cell wall. But we do encounter in the world hypertonic situations, dry, high solute environments, or hypotonic situations like freshwater ponds. Those do create problems. They are what we call osmotic problems, and organisms have to find ways to adapt and survive in those environments if they want to remain there. We call that ability to handle deviations from an isotonic situation. We call that osmoregulation. They can either control solute, they can control water, or perhaps both in order to sustain their life processes in a given environment. We've got an example here. There's a paramecium, usually lives in pond water, which is generally hypotonic. And so this is an animal cell, doesn't have a cell wall. And so water will want to gravitate in. Why? Because inside there you have cytoplasm full of proteins, full of ions. Well, the paramecium has developed this special contractile vacuole. It pumps the water. As water comes into the cell, it gets selectively transported into this vacuole, and then these little channels here are conduits back outside of the cell. It's called a contractile vacuole that basically acts as like a septic pump or a water pump under your house. If you've ever had a heavy rain and you hear that water pump kick in, it's kind of the same basic mechanism. And this is a bit of a repeat slide. That is to say, when we're in a hypotonic situation or close to isotonic, it'll be a turgid plant. And if we're at isotonic or slightly hypertonic, flaccid, the plant wilts. So hypotonic, it'll stay upright. Especially we're talking about non-woody plants here. Even woody plants will do this. It's just that it's a little more subtle. That it'll become flaccid if it's moved into isotonic or especially hypertonic. We've also talked about this a little bit, but I want to make sure you have these definitions. Facilitated diffusion is passive movement of molecules across a plasma membrane. I consider osmosis a form of facilitated diffusion, and I've got some slides to demonstrate that for you. Not everybody will say that, so if you see it out in the book or that sort of a thing, the main difference with osmosis is that if you have a solute gradient, osmosis is going to move opposite of the solute gradient. It's going to move into a higher concentration of solute. That's because it's moving down its concentration. But we facilitate diffusion by creating channels or having carriers to allow molecules to move across the plasma membrane that otherwise wouldn't. So channels, usually limiting by size and ion charge, and then the special channel, aquaporin. Most channels are usually channels for ions. The ions are by far the most common channel protein, but aquaporins for facilitated diffusion of water, otherwise known as osmosis. And here's the evidence for why I believe I consider it facilitated diffusion. What happens in the nephron of a kidney is that you have a filtration step. So we filter here and we have this solution. If it gets down past here, down into the ureter, it will eventually become urine. But up here, it's filtrate. Look at the different colored dots here are different isoforms of aquaporin. Look at all the aquaporins right here, and then bam, nothing. Nothing at all. Water cannot move through this area. What's happening is as you move down this loop of Henle, there's a stronger and stronger osmotic gradient. So water will move out if we let it. So water is moving out, moving out. Now what we do is we trap the water out of this tubule. We're retaining water. We need the water. We live on land. On the way back, though, we're getting more dilute again, and the reason is because we're selectively transporting solutes, in this case sodium, out of these tubules. So we see this very, very limited distribution of aquaporins, where it's water permeable as we're going down, but water impermeant on the way up. And then we have one more water trapping step on the way down to the ureter and then down into the bladder. So this is a water trapping mechanism, facilitated diffusion. With carrier proteins, what we see is either what looks like a revolving door. Sometimes you might see caricatures of revolving doors, or this sort of clamshell-like morphology, where what's going on is they're trying to show you that you have a binding event, a change in the shape of that carrier protein, and then a release. This is just like an enzyme. Think of this molecule as a reactant. We come in here, the reactant gets changed during the conformational shift, and then the reactant is released. So carrier proteins are just like enzymes. They're lowering the energy requirement to get what are otherwise molecules otherwise unable to get across the membrane. Now, if you heated up this membrane, if you added energy to the system, these phospholipids would spread out, and then maybe that molecule could get through. But we don't want that. We want to control it. So what do? We use carrier proteins. And carrier proteins can be affected by things like pharmaceuticals and things. We can use those just like enzymes. In any case, I digress a little bit here. We do see specific diseases caused by transport systems that malfunction, like certain types of kidney disease, what we call cystinuria is an example of that. Cystic fibrosis, that's more of a channel than a carrier. But in any case, we do see diseases associated with malfunction of channels and carriers. Now let's get into active transport. As I mentioned previously, when we're moving something from down its concentration gradient, there's really no need to input energy. The cell is not going to waste energy on processes that will happen on their own if the cell lets it. So simple diffusion, facilitated diffusion. Carriers and channels can facilitate diffusion. Every once in a while, though, we want to move something up its concentration gradient. You'll see that this one here is going from here to here. That requires energy. And only carriers can do this. Channels cannot do active transport. But what happens is that carrier will hydrolyze ATP in order to push, or the phrase is pump, molecules up their concentration gradient. This is a way of energy transfer, it's a way of storage, it's a way of creating systems that will do work for in the cell. And so I've got a few pictures here about active transport. So passive, it works on its own. Like we have enough potential energy that this thing will engage in kinetic energy and complete. This one would not, though, so you need to invest energy in order to get that rock up the hill. But the result is that you have more potential energy at the end. Usually requires some kind of energy, usually in the form of ATP. If we have direct active transport, we call primary active transport. ATP is used in the enzyme complex to do that work. We do have other forms of active transport that are indirect, but still require ATP at some point. It's performed by specific proteins, usually, it's carriers, it is not channels. And so we see that sort of clamshell morphology here, but the roadmaps are this. Look for the gradient. Is it going up its gradient or down its gradient? And if you see ATP, either ATP written on the protein or you see a more complicated depiction of this, like ATP to ADP plus PI, that means energy is being released, and that is active transport. You should be able to spot active transport from a mile away by virtue of the fact that it's either moving against a concentration gradient or it is hydrolyzing ATP. Later on, when you get into more advanced classes, you might talk about secondary or even tertiary active transport. Very, very important processes. But these are passive on the left. Active, meaning that we have to hydrolyze ATP in order to get these molecules to move up their concentration gradients. But among the most important processes in the body, virtually every living nucleated cell in the body has these sodium-potassium pumps. The technical term is sodium-potassium ATPase. It's one type of an active transport system. It's pumping sodium out of the cell and potassium in. But every day of your life, your body metabolizes somewhere on the order of 150 to 160 pounds of ATP. 150 to 160 pounds of ATP. I've been talking to you now for about almost 23 minutes. And in that time, you have burned somewhere on the order of a few pounds of ATP just thinking about this material or thinking about something else, perhaps. I don't know. In any case, what's going on is that sodium-potassium ATPase is pumping 3 sodium ions out and 2 potassium ions in using a molecule of ATP for every time it rotates. And we establish gradients on both sides. So we're really high on potassium on the inside, really high in sodium and chloride on the outside. And we can use those gradients to signal in some cases. The deviations in these gradients is a form of signaling, like in neurons and muscle. We can also use sodium and potassium to do work, moving other things. And so here's the basic mechanism. Again, look for your gradients. Look for your gradients. Step 1 by convention is that we bind 3 sodiums in the low sodium environment. So this is faced into the cell. That's a low sodium environment, so the sodium binds. When the sodium binds, it triggers a phosphorylation step. This carrier has enzymatic activity, meaning that it can take an ATP and transfer that phosphate along with the energy onto itself. This is called autophosphorylation. It's an autophosphorylation step. When that phosphorylation step occurs, it causes the protein to shift and face outward, but it also changes the shape. Look at the shape of the site here. It's big and round, but here it's narrow. That's because that change in shape is actually kicking off those sodiums. They wouldn't release if there wasn't a loss of affinity for those sodiums. Why? Because the concentration's high out here. Okay. If sodium can bind in low concentrations, I guarantee you it will bind in high concentrations. So we have to change the affinity. That's part of that conformational shift. Once that sodium releases, you see now these skinny triangle sites are now wide triangles. That's because now we can bind 2 potassiums. And when those 2 potassiums bind, That causes a dephosphorylation activity in the protein, in the carrier protein. It dephosphorylates, it pops that inorganic phosphate off. It doesn't hook it back up to ATP, pops it off. And that causes the protein to shift back the other way. Again, those sites change, kicking off those potassiums, and now we have those big, beautiful, round sodium sites for binding, and we repeat the cycle. Sometimes you will see this as a revolving door, but some of the key things: 3 sodiums for every 2 potassiums. We have this autophosphorylation and this auto-dephosphorylation. Conformation change, binding, conformation change, release. Binding, conformation change, release. Several themes here that you should get to know because you're going to see these things again, certainly later on in physiology, probably in cell biology, probably in biochemistry if you take it, or other biology courses. Very, very important. This, by the way, is a major target for certain types of anesthesia. So if you're interested in orthopedic surgery, or you're interested in working with patients from orthopedic surgery, this will become a major aspect of the care that you give to those patients. And just some more pictures here. Can you label these? Can you label these in terms of channels and carriers? Can you describe these in your own words? That's something I think that we don't do enough of, is getting you to try to deal with these things in your own words. Yeah, you can repeat the words, and that's okay. But the more you can do this in your own words and describe it to somebody else, the better you understand it. And I think that's a good thing to do, is to be able to convey this to others. We're going to finish off a few slides talking about vesicular transport. Vesicular transport is really, really complicated. There's a lot going on here. Well, we're going to try to summarize it a little bit. What you do is you're using a combination of proteins and the plasma membrane. And we call this cytosis. Like, for instance, if we're ejecting material into the extracellular space, extra outside the cell, it would be exocytosis. If we're creating a vesicle, bringing stuff in, we call that endocytosis. And the in-betweens are vesicles. Normally, when we use channels and carriers, we're talking about water, we're talking about small molecules with big polysaccharides, big proteins, or large amounts of certain things like fat droplets. We bring them in by endocytosis. If we're releasing large amounts of things like neurotransmitter, or in some cases hormones, we'll release that vesicle, and that's exocytosis. But it's a combination of using the plasma membrane and proteins. It's all active transport, so it's always using ATP, and it's complex. It's bulk transport processes. And so exocytosis, what we do is we bleb off a membrane vesicle, and it fuses with the plasma membrane. That would be a form of secretion. Some secretion happens continuously in some things. Other forms of secretion are regulated. You have some sort of a signal that then tells the Golgi, hey, start releasing vesicles. Hormone signals are a lot like this. Try to think of an example of this. Nothing's coming right to mind, but maybe maintenance of plasma membrane in some cases would be what we would call constitutive. On the other side, what we do is we invaginate, that's the phrase, invaginate the plasma membrane in order to create a new vesicle. So this pinches off here, and now we have this vesicle with this aqueous environment inside. That can be solid particles when we talk about phagocytosis. One of the most common things we do with phagocytosis is ingest bacteria or cellular debris. Very, very common in the human body. Pinocytosis, that's more common in digestion. Like, for instance, fat droplets are brought into actually the lymphatic system in your digestive tract using pinocytosis. Then there's this third one that we call receptor-mediated endocytosis. That's because they observe these special proteins called clathrin and other things. Here's the thing. These don't really just happen on their own without a receptor and some sort of a signal. So while they call this receptor-mediated endocytosis because they've spotted the receptors, the truth is all these things have receptors. So I kind of get annoyed by that. Phagocytosis are solid particles. Pinocytosis are usually liquid droplets, fat droplets in particular in human systems. Receptor-mediated endocytosis can be any number of things. It can be small molecules, big molecules. It just depends on what the receptor is sensitive for. When those receptors are activated, then you start to get invagination and you create this vesicle. But it's endocytosis. Some great videos out there. Once you can dial these up for a few minutes of entertainment to kind of see, for instance, phagocytosis process, pinocytosis. What they're talking about here is that when you bring in a solid particle, generally what's happening is that you're going to fuse with things like lysosomes in order to break down whatever inside that particle. I think that's also probably true with pinocytosis as well. Main thing is that with receptor endocytosis is they've identified the receptor, and these things don't have to be big, huge particles. They can be relatively small. It's very specific. So we have this thing called a ligand, or sometimes people say ligand. It binds a receptor and it triggers the motion of the plasma membrane to begin creating a vesicle. But the main hallmark of receptor-mediated endocytosis versus the other two is that you have this protein that forms on the outside of the vesicle. It's a coat protein. The name of the protein is clathrin. Sometimes they'll say a clathrin-coated pit. But what we have going on here is that the molecules aren't as big and it's much more specific. Okay, much more specific, but it's the same story as these. Like I said, these guys have to have receptors too. Okay. So that completes chapter 7. So make sure you're taking good notes. I know that can feel like a hassle, but I would recommend you write stuff down. You draw pictures. You use your muscles. Yeah, you got to use your brain muscle, but use your brain muscle in ways that more than just trying to be a sponge, because the brain isn't actually a very good sponge. It works better when you exercise it, doing different modalities of learning, and one of those is taking good notes, writing this stuff down, doodling some of these pictures and things to help your brain wrap around the knowledge. But thanks for your time. We'll come back later on with chapter 8. So we'll talk to you soon. Take care.